Thank you again for joining us today to the Become an RNAscope Expert webinar series hosted by the ACD Technical Support Team. Today's speaker is Dr. Robert Monroe, the Chief Medical Officer at ACD, who is a board-certified pathologist with extensive experience in situ hybridization and immunohistochemistry. In today's webinar, Dr. Monroe will discuss data interpretation with RNAscope. Before we begin, let us go over today's webinar rules. The audience will be automatically muted. If you would like to ask a question, please select the chat button to ask your questions. We may not get to all your questions, however, but we will contact you directly after the webinar to address your questions in the event that we miss you. Welcome again, and we'll hand over the stage to Dr. Monroe.
Thank you, Jackie, and thank you to all the audience members who've taken time out of their busy schedules to join us this morning for this webinar on RNAscope data interpretation. The agenda for the webinar will be as follows. We'll review RNAscope as well as some of the main RNAscope applications, and then we'll dive into a more detailed review of RNAscope staining and scoring, and then finish up with an overview of the quality control procedures that we typically recommend for RNAscope results in a Q&A session. So with that, I'd like to begin with a brief overview of the RNAscope technology. Really, IHC and FISH have been the leading tools for research and tissue diagnostics over the past 3 to 4 decades, with IHC technology dating back to the 1970s, along with DNA FISH technology really coming into its own in the 1990s. There was no sensitive or robust technology for RNA in situ hybridization or detection prior to the development of the RNAscope technology, which was launched as a product back in 2010. The RNAscope platform, this slide will just provide a brief overview of the overall platform. It starts with a unique probe design for each target, relying on the so-called double Z probe strategy to ensure maximum specificity. The probe design, in combination with the amplification steps of the assay, provide for a roughly 400-fold amplification of the signal, with concurrent suppression of any nonspecific background staining in the assay. The sensitivity of the assay allows for detection of all the way down to single RNA molecules in single cells, which are visualized as punctate dots in their morphological context. And these dots can be easily visualized with standard microscopes, either light microscopes or fluorescent microscopes, and then quantified through manual scoring systems or image analysis applications, either of which are suitable for this type of analysis and can allow for quantification of the RNAscope signals. Returning to the probe design, the technology is based on the use of Z-shaped oligonucleotides composed of a lower region of approximately 25 base pairs, which are connected through a spacer region to an upper tail region shown here. When a pair of Z probes hybridize next to each other on a target-specific region of an RNA molecule, this region being about 50 base pairs in length, a so-called pre-amplifier binding site is created with the upper regions of the Z probes. The target probe for a given biomarker consists of approximately 20 double Z pairs, which span a target region shown here of about 1,000 base pairs. This double Z probe design is the foundation for the highly sensitive and specific messenger RNA detection technology that is one of the hallmarks of RNAscope. The steps following hybridization of the double Z probes that we just visualized along the target sequence provide for signal amplification and further specificity of the assay. The preamplifier molecule shown here which is shaped like an L, will only bind if it encounters a pair of adjacent Z probes, which create that unique preamplifier binding site. In subsequent amplification steps, the hockey stick-shaped amplifier targets, shown here, and circular label probes bind to the preamplifier and amplifier targets respectively, leading to an approximate 400-fold amplification for every Z pair. If a Z pair happens to bind to a non-specific target region, as shown here, the preamplifier, again the L-shaped probe, will not bind given that there's no specific preamplifier binding site, as there's not a second Z probe here, and no signal is generated in this situation. Stepping back, overall this 1 kilobase target region for this amplification strategy for a roughly 1 kilobase target region has the potential to recruit approximately 800 labeled molecules, which is the product of 20 Z pairs multiplied by 20 amplifiers multiplied by 20 label probes. So the double Z strategy that we've been discussing really derives its robustness and reproducibility for detection of RNA through the redundancy of the double Z probes. Although most RNA target probes utilize on the order of 20 double Z pairs, hybridization of only 3 of these double Z pairs, 3 out of 20, is required for detection of that single messenger RNA molecule. This redundancy allows for detection of fragmented targets in the context of formalin-fixed paraffin-embedded tissues, which are known to contain many RNA double-strand breaks, for example. Other fixation methods can also interfere or disrupt RNA. Proteins bound to RNA also can be an obstacle to the accessibility of the Z probes to binding, and this strategy, which again requires only roughly 3 out of 20 Z probe pairs to detect a single molecule, ensures that these molecules can be detected in the presence of some protein binding. While the details of the performance of the assay are beyond the scope of the current webinar, RNAscope can be performed either manually with the help of a hybridization oven available through ACD, or also on automated platforms. And ACD has designed specific kits as well as optimizing protocols for both Ventana and Leica automated staining platforms. At the present, automation is only available on the Ventana and Leica research platforms, the Ventana Discovery as well as the Leica RX systems. But in the latter part of this year, select RNAscope assays will actually be available on Leica's clinical platform, the Bond 3 system. Overall, automation has the potential to not only save hands-on time for operators and laboratories, but also to provide for greater reproducibility in RNAscope assay performance from day to day as well as operator to operator. Through February of this year, the RNAscope technology had been used in over 400 publications spanning most of the top journals, many of which are shown here, including Nature, New England Journal of Medicine, Cell, and so forth. Publications have really spanned the spectrum of research applications, including the fields of cancer, infectious disease, inflammation, neuroscience, and other major areas of investigation. Before diving into the details of the scoring, that it's really the heart of this webinar, I thought it would be instructive to review a few of the many applications RNAscope fulfills and that have been described in some of these publications. The first of these applications that I wanted to highlight today is an application for HIV research. And this example, and the example shown in this slide, noted HIV expert and researcher Dr. Jake Estes of the National Cancer Institute compares RNAscope shown on the left, against the previously standard non-isotopic in situ hybridization methods that have been commonly practiced over the last 10 to 15 years, which utilize digoxygenin probe labeling. And in this case, Dr. Estes was attempting to detect simian immunodeficiency virus in lymph node tissue. As you can see, the RNAscope signal on the left is roughly 100 times stronger than the standard signal seen in the non-isotopic preparation on the right, highlighting the great improvement in the sensitivity of the technology over traditional methods. Another application of the technology is for detection of intratumoral heterogeneity for the presence of specific biomarkers and genetic changes. In this prostate cancer study, dual RNAscope staining was performed for the ERG and ETV targets, the ERG shown in brown and the ETV target shown in red. Both of these targets are overexpressed in prostate cancer as a result of gene fusion events that take place in many prostate cancers. And they show differential and mutually exclusive expression patterns, with the left-hand panel showing ERG expression in this loci, while in the right-hand panel, the ETV expression is shown in a separate location of the tumor. These are both mutually exclusive of each other, suggesting that these independent events lead to differential expression patterns in prostate cancer. This combination of molecular detection and morphological context provided by this duplex RNAscope assay is really not possible with traditional grind and bind, next generation sequencing, or PCR assays, which destroy morphology. They're also not available with standard IHC assays, which really don't provide the genomic information provided by RNAscope. RNAscope can also be employed in both chromogenic assays, as shown in the previous two examples, as well as fluorescent assays, as shown here. In this example of circulating tumor cell detection in a breast cancer patient, RNAscope target probes for CD45, bone marrow-derived cells shown in green, and for pan-keratin positive circulating tumor cells. Actually, correct that the pan-keratin positive tumor cells are shown in green, and the CD45 bone marrow-derived hematolymphoid cells are shown in red. Similar to what is seen in chromogenic assays, the CD45 positive cells show punctate red dots, seen in this enlarged image in the upper right-hand panel, while the circulating tumor cell is highlighted by these clusters as well as individual dots shown in green. It goes without saying that immuno-oncology is one of the currently most exciting areas of research, and RNAscope is a great tool for the identification of specific cell types of interest within tumors expressing various immuno-oncology markers, including PD-L1 and other cell surface receptors and cytokines. In this example of a dual RNAscope assay for PD-L1 and CD8 in a non-small cell lung carcinoma, the green signals represent PD-L1 expression by lung cancer cells, shown with the green arrows, whereas the red signals represent extensive infiltration of CD8 positive infiltrating cytotoxic T lymphocytes in the stroma around the tumor. Occasional co-expression of both PD-L1 in blue as well as CD8 in red suggests the presence of a small subset of cytotoxic T cells with PD-L1 expression. The last application that I wanted to highlight was an application in the field of so-called head and neck cancers, where RNAscope assays for the human papilloma virus are proving to be very valuable for the characterization of squamous cell carcinomas. Numerous studies utilizing the RNAscope technology have shown, as well as other markers, have shown a difference in the behavior and aggressiveness of HPV-related squamous cell carcinomas relative to those non-HPV-related tumors, which are thought to be related to smoking and or alcohol exposure. In comparison to these other assays, such as DNA in situ hybridization for HPV, or P16 IHC with P16 representing a surrogate marker, a protein that is expressed downstream of HPV infection. The RNAscope assay has shown superior performance with higher sensitivity than DNA-ish and higher specificity than P16 IHC. Furthermore, the HPV RNA assay shows direct evidence of viral gene expression, which is really something that many of the investigators involved in these studies are hoping to see. Moving on to RNAscope scoring, let's start with a quick comparison of RNAscope and immunohistochemistry. Both can be performed in chromogenic or fluorescent assays. Similarly, both can be performed on formalin-fixed paraffin-embedded tissue, the predominant type of tissue used in research, as well as other types of tissues fixed with other modalities. The signal observed for most IHC studies is a diffuse signal, whereas the signal seen in RNAscope studies consists of dots and clusters. The localization of the protein detected by IHC varies depending on the protein target, whereas RNAscope localization, the localization of the RNAscope signals is uniform in most RNAscope studies and is seen in both the nucleus as well as the cytoplasm. For cell types, both IHC and RNAscope are effective in detecting targets in almost any cell type. Some of the common IHC staining patterns are shown and listed here, including nuclear, cytoplasmic, membrane, a combination of nuclear and cytoplasmic, as well as a combination of cytoplasmic and membrane staining in the lower right-hand panel. Other staining patterns are also possible but less commonly seen, and these include staining of specific subcellular compartments, organelles, or specific areas of the cell. In comparison to this diffuse staining pattern of most IHC assays, such as the current example of a P16 stain, which we had previously seen, exhibits both a nuclear and cytoplasmic diffuse staining pattern. The RNAscope assays show dots and clusters in both the nuclear, as seen here, as well as the cytoplasmic compartments. You can see the dots surrounding the nucleus as well as in the nucleus. As we go through the next few slides, I'd like to provide some additional perspective on some of these terms that I'm using to describe RNAscope staining and to address the following questions, including what does a dot mean? What is the significance of dot size? What is the difference between a dot and a cluster? And finally, what is the sensitivity of RNAscope versus immunohistochemistry? So starting with the difference between a dot and a cluster, the differences are highlighted in this current image. Single messenger RNA molecules are detected, as shown here, as single punctate brown dots, while larger dots, for example, shown here, which can also be referred to as clusters, result from overlapping signals from multiple messenger RNA molecules. The variation in dot intensity, so for example, if you compare this dot, which has a slightly less intense brown color than an adjacent dot here, this variation in dot intensity can also be related to differences in the number of double Z probes bound to a target molecule. Similar patterns of dots and clusters can be seen in fluorescent versions of the assay, again with the dots, single dots shown here and clusters of dots shown with the upper arrows. Moving on to the detection limit for RNAscope. This detection limit is typically below the detection limit for IHC. In this example, comparing RNAscope and IHC detection of the biomarker c-MET, you can see that the low expressing c-MET cell line shown in the top row has no detectable immunohistochemistry signal. However, there are clearly positive RNAscope dots present in the RNAscope assay for c-MET, which are above and beyond the negative control run in parallel with the target-specific c-MET probe, indicating that this low level of expression, these low numbers of dots, are true mRNA signals. So this really demonstrates the ability of RNAscope to pick up low level of expression for messenger RNA not typically seen by immunohistochemistry. In comparison to RT-PCR, RNAscope shows a similar sensitivity and limit of detection. In the two panels shown here, starting with the experiments highlighted by the figure on the left, which looks at the expression of HER2 in a spectrum of breast cancer tissues, the number of dots per cell, shown on the x-axis, was highly correlated with transcript levels, shown on the y-axis, as determined by reverse transcription PCR. Similarly, for quantitation of the target NRG1 in cell pellets representing various dilutions of NRG1 expressing cells, the number of dots shown on the, in this case, the y-axis, was highly correlated with the quantitative PCR on the x-axis, showing a near slope of 1. Moving on to scoring, there are a number of ways to score RNAscope assays, ranging from qualitative to semi-quantitative to H-scoring and image analysis, all of which we'll cover in some detail. For some assays, particularly those looking at markers that are highly expressed when present, simple qualitative scoring can be appropriate. The panel in this slide shows clearly positive expression of the biomarker albumin by RNAscope on the right, with clearly negative DAP-B expression on the left. So this is an example where simple qualitative staining is sufficient for the application in question, which is looking for the presence of albumin in hepatocellular carcinoma. Semi-quantitative scoring systems are quite common for immunohistochemistry assays, and one of the more commonly used systems is shown here with a scoring range from 0 to 3+, with corresponding staining intensities ranging from negative to strong. This system is similar to what is used by many research as well as clinical diagnostic labs for a variety of IHC markers, including HER2. Similar semi-quantitative scoring systems have been developed and are commonly used for RNAscope assessment, both by ACD as well as many of ACD's clients. The scoring schema shown in this slide is what is routinely used by ACD and its Pharma Assay Services group, and shows a system that spans a scoring range of 0 to 4, corresponding to the numbers of dots per cells, along with some information on the presence and number of clusters. A case with a score of 0 has essentially no staining, or less than 1 dot for every 10 cells analyzed. A score of 1 correlates with the presence of 1 to 3 dots per cell, while a score of 2 correlates to 4 to 9 dots per cell, and so on. Some examples of this scoring schema are shown in the following panels, with the first panel shown here showing a semi-quantitative score of 0, obviously showing, in this case, essentially no staining. An example of a score 1 correlating with the presence of 1 to 3 dots per cell is shown in the following two panels. So you can see the epithelium in the left-hand panel showing a few dots per cell, and a prostate carcinoma specimen in the right-hand panel, again showing approximately 1 to 3 dots per cell. An example of a score 2 in this RNAscope scoring schema is shown here. Similar epithelium on the left shows slightly more dots per cell than was seen in the last panel, approximately 4 to 9. Similarly, on the right, you see an increased number of dots per cell in the range of 4 to 9, and so forth. I won't go into detail on all of these, but you can see gradually as the score increases, the number of dots per nucleus increases, and you begin to see some of these clusters, the darker dots representing overlapping signals. Finally, an example of a score 4 in this scoring schema where most of the cells have greater than 15 dots, and within those cells, greater than 10% of the dots are present in clusters. So you can see numerous clusters in these cells, and an example here of a brown chromogenic assay. As for IHC, H-scoring is also compatible with RNAscope. For those of you who are not familiar with H-scoring, it's basically just a method of assessing the extent of staining for a biomarker in a given tissue that incorporates both staining intensity as well as the percentage of cells with given staining intensities. For IHC, the H-score is obtained by the formula in this case, 3 multiplied by the percentage of strongly staining cells, plus 2 times the percentage of moderately staining cells, plus 1 times the percentage of weakly staining cells. And this provides a score range from 0 to 300. For RNAscope, staining categories are determined by the number of dots per cell, as we've just gone over, rather than the staining intensity. But otherwise, the H-scoring system is highly similar. If the score range from 0 to 300 is insufficient and additional categories are desired, the maximum H-score increases accordingly, according to the formula shown here, the number of staining categories minus 1 multiplied by 100. Moving on to image analysis and quantitative scoring. ACD has and currently uses 2 main different software applications for quantification of RNAscope assays. There are other software applications available for so-called dot counting in assays like RNAscope and FISH, but the 2 that ACD typically uses are called SPOTSTUDIO and HALO. SPOTSTUDIO is a custom-designed algorithm developed by ACD in partnership with the company Definiens that enables quantitative analysis of chromogenic single-color RNAscope assays captured using whole slide scanning systems. HALO is a second software application offered by Indica Labs as well as through ACD's Pharma Assay Services group that provides a very similar quantitative analysis for single-plex assays, but also provides the ability to analyze more complex duplex as well as multiplex RNAscope assays. An example of SPOTSTUDIO image analysis, in this case for HER2 expression in breast carcinoma, is demonstrated in this slide. The software basically identifies cell boundaries shown by the solid lines, sort of the chicken wire appearance, by approximating the average distances between the cell nuclei shown with the hematoxylin stain. The algorithm then counts the dots within each cell and bins the cells by color according to the number of dots per cell. So in this case, the analysis shows blue cells containing roughly 10 blue cells containing 0 to 1 dots. The blue cells shown here. Approximately 32 cells indicated in green with between 1 and 3 dots per cell, and so forth. Approximately 30 red cells containing between 3 and 6 dots per cell, and approximately 25 orange cells containing greater than 6 dots per cell. The HALO software can perform highly similar analyses to those seen in the previous SPOTSTUDIO analysis on singleplex assays, but allows for more complex duplex analyses as well as H-scoring. In HALO's quantitative H-score analysis shown here, each cell is assigned to a bin based on the number of dots per cell, again similar to the SPOTSTUDIO example that we just went through. Clusters are divided by the typical probe signal area to calculate a dot number for a given cluster. And then an H-score calculation is performed to assign a single score for a selected area, for example, this square panel, based on the average target expression in this tissue. So moving on to the RNAscope control strategy. For proper interpretation, these assays should always be performed with concurrent positive and negative controls to be run in parallel with the target probes of interest. These positive controls are typically housekeeping genes that are expressed at similar levels in most tissues, and so can provide a baseline level of expression to which the target probe can be compared. The common positive controls utilized in RNAscope assays include UBC, POL2RA, and PPIB, and their use is really analogous to the use of genes like beta-actin, GAPDH, and other similar types of housekeeping genes in RT-PCR and northern blot experiments. The purpose of the positive controls is to demonstrate the integrity of RNA for RNAscope testing. In general, if a signal is present in the positive control slide, the tissue is suitable for RNAscope, whereas if the signal is absent, the tissue is not suitable for RNAscope. The negative control that's typically used for RNAscope assays is a probe targeting a bacterial gene called DAP-B. DAP-B is used to assess nonspecific staining and nonspecific signals. And in general, if signals are present, tissue is not suitable for RNAscope, indicating that there is a high degree of background staining. Whereas if the signal is absent, indicating that no nonspecific or essentially no nonspecific staining is seen, the tissue would be suitable for RNAscope. Depending on the target, different positive control probes are recommended. And this table summarizes these recommendations. For example, UBC, which has a medium to high expression level, should be used with high expression targets but not low expression targets. Because of this high level of expression seen with UBC, the signal may be picked up even in the presence of significant RNA degradation, and therefore, if it's used to qualify tissues for target probes, it may lead to false negative target results. PPIB, shown in the middle row, has a moderate level of expression and is recommended for use as a positive control for most tissues. Its expression level is low enough to provide a rigorous control for RNA quality for most samples. In the majority of studies, if PPIB is positive, then any target probe will detect the presence of target RNA. And so this is the probe that we recommend for most studies. Finally, POL2RA has a low expression level and is recommended for use with low expression targets. Prior to running an RNAscope experiment with target probes, the technique of a lab in running RNAscope should be checked and samples should be qualified with the appropriate positive and negative controls to make sure everything is working correctly before investing a lot of time and effort into staining and performing experiments with the target probe. In the first step, slides prepared from cell lines are tested with appropriate positive and negative controls. If the PPIB signal, shown in the bottom panel, is robust with at least 2 dots per cell or more, and the DAP-B signal is essentially negative with less than 1 dot per cell, the technical check passes, this first step passes, and one can proceed to the sample RNA quality check. In this step, the sample tissue, typically FFPE tissue, is probed again with the same positive and negative controls, in this case DAP-B and PPIB. And similarly, if the PPIB signal, the positive control signal, is sufficient with at least 2 or more dots per cell, and the DAP-B signal is negative with less than 1 dot per cell, one can proceed to running target probes and consider the tissue in question for target probe analysis to pass all QC. If for some reason the PPIB signal is weak or less than 2 dots per cell, or the DAP-B signal suggests nonspecific staining, that would be considered a failure of that particular tissue, in which case the lab or the operator should go back and verify technique, look into the RNA quality of the tissue sample, and potentially perform some assay optimization. The following example provides a step-by-step review of this QC process, in this case on an HPV target probe assay. In this first step, corresponding to the step 1 that we just went over in the QC slide, a technique check with cell line controls is performed and shows abundant UBC staining. So in this case, UBC was selected as the positive control, and absent DAP-B staining in the negative control. These are HeLa cell pellets. So in step 1, the QC passed with, again, plenty of positive control staining and essentially absent DAP-B staining. In step 2 of this example case, the tissue sample is checked for RNA quality with the application, again, of the same positive and negative controls. So whereas in the first step, HeLa cell lines were tested with the positive and negative controls to make sure that the lab had the technique worked out, in the second step, the actual tissue of interest is qualified or is attempted to be qualified through the application of the same probes. So in this case, the UBC probe shows the presence of signals, whereas the DAP-B probe shows absent signals, again demonstrating quality control check. And this essentially qualifies the tissue for proceeding forward with RNAscope testing using the target probes. In step 3 of this example case, the tissue sample, having been qualified by the positive and negative control probes, is tested with the actual target probes of interest. In this case, the target probes of interest being various HPV subtypes, including HPV-16, HPV-18, a cocktail of HPV-31 and 33, and a cocktail of HPV-6 and 11. The three left-hand panels are all high-risk HPV subtypes, whereas the right-hand panel is a cocktail of low-risk HPV subtypes. As you can see, only HPV-16 is positive for RNAscope expression in this among this set of target probes, indicating that the HPV infection involving this particular head and neck carcinoma is tied to or is linked to the high-risk subtype HPV-16. An optional step 4 can also be performed and is recommended if known positive and negative control cell lines or tissues are available, similar to IHC control strategies. In this case, a cell line known to be positive for HPV-16, the CASCI cell line, shows expression of HPV-16, whereas a cell line known to be negative for HPV-16 shows absent expression. This step is recommended if known positive and negative control cell lines or tissues are available, again similar to the strategies employed for many IHC assays. Once the technique is working reproducibly in a lab for RNAscope, labs can choose to skip step 1, again, which is the technical check, and run steps 2 and 3, which consist of a tissue sample check and test probes concurrently. In this RNAscope experiment, the positive control PPIB shows robust, moderate levels of expression, while the negative control, again, the DAP-B, shows essentially no signal. The test probe for the EBV, is strongly positive. You'll note in this case that the signal actually is similar to what's seen in some immunohistochemistry assays. Essentially, the dots and clusters have become confluent because the level of expression of EBV in these cells is so high that the dots and clusters can't be discerned individually. For most labs, once RNAscope testing becomes routine, particularly if automation is available, the sky is really the limit. With a variety of applications possible across any species, any gene, and any target. And so with that, I'd like to open up the webinar to a Q&A session to address any questions the audience may have. So the first question I'd like to address is as follows. The question is, sometimes it is hard to make out the boundaries of the cells after RNAscope processing. How do I normalize the signal? So this is essentially a two-part question. The boundaries of the cells can be affected by both the fixation method as well as the pre-treatment conditions of the assay. And playing with the pre-treatment conditions can allow for better visualization of the nuclei. So for example, if the pre-treatment steps are reduced in time, the nuclei and therefore the cell boundaries are easier to visualize, for example. And so that's one of the ways that allows for determining whether the dots that are seen belong to one cell versus an adjacent neighboring cell. The second part of the question, how do I normalize the signal? Normalization can be done if there's sufficient interest by comparing the number of dots seen with the target probe to the number of dots seen with the positive control probe, which typically is either the UBC probe or the PPIB probe. So that expression level seen in the positive control probe can be used as a way to normalize expression across different tissues that may have been obtained at different times or stained in different experiments. The next question is as follows. Sometimes very faint dots are present. How do we know if these signals are real? What is the threshold to determine if a dot is real or not? That's a great question, and the faint dots typically would be compared to the presence of similar dots in the negative control slide that's prepared in parallel to the target probe, the DAP-B slide. If those faint dots are present in the DAP-B slide, then they're likely representative of background staining. However, if they're absent, those faint dots that are seen in the target probe slide may well represent low level of expression in that of the target probe in that target tissue. And the recommendation would be to attempt to optimize the protocol to make those dots brighter to see if in fact they really do represent true expression. And there are a variety of ways that the intensity of those dots can be increased by decreasing the fixation time, for example, or altering the pretreatment conditions. And we can work with you on troubleshooting through our customer service group if that's something that would be of interest to the person submitting that question. The next question is as follows. Samples such as melanoma, where a lot of melanin is present, are sometimes probed with RNAscope assays. What would you recommend to discriminate between real and fake signal? Melanin is a pigment that's typically brown in color. That's very similar to the brown chromogenic clusters that are seen in higher expressing RNAscope targets. I would recommend, in the case of melanoma research, to look into using an alternative chromogen. We typically would use a red chromogen, which is fairly easy to distinguish from the brown melanin pigment. So I think that would be a possible solution for that issue to help you discriminate between the endogenous melanin and true RNAscope signal. Question of access to the slides provided in this webinar. The webinar will be posted on ACD's website, so there will be the ability to go back and access these slides. Another question is as follows. Is it possible to combine the RNAscope technique with IHC staining, for example, with a cell marker, potentially a cell surface marker? The answer is yes. Many of our customers have been successful in running dual IHC and RNAscope assays using different chromogens, but they've had excellent results in using the two modalities together. And we can point you to some papers and provide some advice on how to approach those dual assays if you, again, contact our customer service group. Another question has been submitted as follows. What about alternatively spliced mRNA molecules? How do you test or differentiate between these different mature forms? That's another excellent question. The standard RNAscope technology that's available through the ACD website typically does not have the sensitivity to detect small differences in the splice junction seen in these alternative splice variants. However, a next generation of the technology is being developed by ACD to allow for that type of discrimination, where a smaller number of base pairs that are different between one splice variant and another would be it would be possible to detect with this higher sensitivity assay. And so that's something that ACD hopes to make available through its Pharma Assay Services group. In fact, it already has that available through its Pharma Assay Services group, and it may be available as a product at some point later in the year. But until that time, if there are interests in that type of higher sensitivity, our internal Pharma Assay Services group can perform those experiments. Moving down the list, if all the dots are in clusters within cells, is it proper to score the staining? That's an excellent question. It generally makes it more difficult to count the dots, obviously, if they're all present in clusters. But I would recommend a semi-quantitative scoring system for those. And if you look back at the typical scoring system that we use in our Pharma Assay Services group, a lot of those clusters, if they're present in greater than 10%, those would correlate to a score of 3 or 4, depending on the precise percentage of those clusters in that system. And so I would recommend a semi-quantitative scoring system. But those, for a variety of reasons, we've seen that, and it's not always clear why it happens, but it's something that we've seen before, where there's more clusters and fewer dots. The question, another question that's been submitted, would you expect to get the same or similar results of quantitation when using same probes and sections but with a different kit, say red versus brown, for example? Typically, the signal amplification, the ability to detect individual messenger RNA molecules, is independent of the chromogen used. And so the results should be highly similar whether a red or a brown chromogen is used. How short of an mRNA can RNAscope detect? Again, an excellent question. So we described the typical scenario of roughly 20 double Z probes being the ideal number to detect a single messenger RNA molecule, or being designed as for a target probe to detect down to the single molecule level. But we know that as few as 3 double Z pairs can lead to the detection of a single molecule. So given that, we typically like to have at least a couple of 100 base pairs and at least 10 to 15 double Z probes in our probe design to ensure that that target can be detected in the presence of some RNA degradation. So while you may be able to go to a smaller target size or a smaller number of double Z pairs, we like to use at least 10 to 15 in our standard assay design.
Thank you all for attending today's webinar. This concludes our webinar series for today. If we did not get to your questions, we'll be reaching out to you directly. We will contact you through email. Thank you so much for attending.